the second part of fundamentals of QuPath for image analysis. So this will be building on the part one that I think we did in May. Now I'm recording this, so we'll add this to our YouTube channel for if you want to watch it later. Now, this will be mostly going over training the machine learning algorithms that are inbuilt into QuPath. So just remember that this is presented by the bioimaging platform. My name is Chad Johnson. I'm the optical microscopist in the platform. So we also offer electron microscopy services, histology, and flow cytometry, and you can contact us on bioimaging@latrobe.edu.au. We're on multiple social media platforms, Twitter, Blue Sky, LinkedIn, so please follow us. And as mentioned before, we have started a YouTube channel just to contain these workshops, as well as uploading help videos for frequently asked questions. So those will, I'm trying to get those up, but this is basically a repository for our workshops. Cinema rules. So if you have any questions, please put them in the chat. If you have any larger questions, then feel free to email me afterwards, and we can discuss them then. So the aim of this workshop is to continue learning about the functionalities of QuPath. We will be mainly training machine learning models for automatic detection of multiplexed imaging. And it's also to get an idea of the vast amount of processing and analysis capabilities that the software has to offer. These sort of examples are very specific, but they can be applied to other data sets, and the functionality that we'll be using can be applied to a whole host of other problems. Plus, QuPath has a lot of inbuilt plugins that you can use, as well as ones that aren't inherently in them. You can download them and add them for extra functionality. So, brief overview. We'll be going over a quick refresher on some of the functionality within QuPath, just going over very, very quickly what we did last time. Then we'll be moving on to the main topic, which will be machine learning. So we'll be mainly discussing the object classifier as well as the pixel classifier to some degree. Now, I've booked in 2 hours for this. I don't think it's going to take that entire amount of time, but depending on questions, etc., and how waylaid we get, we'll try and finish a bit earlier. So QuPath refresher. So I'm just going to put that to the side. So QuPath can be loaded very, very quickly. So you may notice that when you install QuPath, you'll get a QuPath standard as well as a QuPath console. So I always use the standard QuPath for practically everything. The QuPath console has a console which allows you to use other functionality using the command line function. I think at this sort of stage, I mean, even I would never use them. I'm not of that level to be able to use that functionality, but using just QuPath as is, it is usually perfectly fine. Where's that gone? Okay, so just very, very briefly, so over here is our project pane, our how we keep track of things within our project. So you can open up individual images within QuPath, but it can be easier to build a project, especially if you're building up a model. That way you can apply the algorithms to other images within that project. If you're opening up a single image, you can basically only work with that image and can't really apply anything else. So it's best to create a project. So what we're going to do is we're going to go create project. And of course, like I'm usually, when I do these, when I attend these workshops, not as a presenter, I typically just watch. You're more than welcome to work along with me, but I'm usually a watch and then do the things later. Depends how good you are at multitasking. So I'm going to go into the folder that that you should have access to, which is QuPath Workshop 2025. And I'm going to actually no. So I'm going to create my QuPath project. I usually have a folder called QuPath in my documents folder, and I'm going to create a new folder. You cannot create a new project within a folder that already has a project in it. It's one project per folder. So I'm going to go to QuPath Workshop 2025 Part 2. And I'm going to select that. And now I have essentially a place to put all of my images, create my objects, create my models, etc., and they can all be, you can add as many images to it as you want, which are called off specific locations in your computer or on your network. Now, if you move the images to a different place, QuPath will lose track of them, so you have to redirect QuPath to where those images are. Now I have my project, so I'm going to add some images. So I can click images, or you can just literally just drag and drop them into this pane. So import images to the project. I'm going to click choose file. So you can drag them, drop them in here as well, or just choose the files. Now I'm going to go into that shared folder that you should have access to. So QuPath, where is it? QuPath. Where did I lose it? QuPath 2025, workshop data. Now I'm going to add, we did the HDAB image last time, so I'm going to add these images. These are 7-channel fluorescent multiplexed images. So I'm going to add that to my database, and I'm going to import. Now, the default image server, usually I think it uses the JSON server or the OpenSite, I can't remember. It doesn't really matter. This is literally just how the images is going to be decoded and read. I know that these are fluorescent images, so I'm just going to say that it is fluorescent. I'm not going to rotate them. And I'm going to leave everything else as is and import the images. Now the images are being imported into, they're being left where they are, but they're actually being directed into here so that we can do whatever we want to. The images aren't edited while we're working with them. The changes are strictly within the project. The images themselves are left alone. So I'm going to click on this image here, which is the image that we're going to be using to train our model. Now, this is a 7-channel image with a whole bunch of cancer markers. So we're going to be training QuPath to detect cells which are positive for each one of these labels. You can show and hide the project pane by clicking on this button here if you need to. These are all your ROIs, some of which we will be using. This is the move button if you want to move things. You can draw boxes, you can draw circles. These ROIs are used to select specific parts of the image. You've got your polygon tool there. The paint tool can be quite handy for selecting large areas. The only thing is you have to color it in. Now, the size of that paint tool depends on the zoom. So if I zoom in really close, it gets very, very specific. Unlike Fiji, the ROIs are automatically added to our annotation manager. In Fiji, it's called the ROI manager, but in here it's called the annotation manager. Basically the same thing, just areas that we're interested in, and you can select and change these as you wish. Select and press the delete button if you want to get rid of them. This button is very important. This is our brightness and contrast dialog. It also allows us to keep track of our channels. So these are all the channels that we have in this particular image. These are specific multiplexed opal stains that allow us to stain individual cell types. You can turn them on and off as you would like. Down the bottom, if you select one, you can use the display curve to change the intensities of these depending on what you need. If you want to reduce a bit of the background, if you want to amplify the signal. Just to remember, the display curve does not change the data. The intensities, the actual pixel intensities remain the same. It's just literally changing how we see it. So even if I change it, this purple channel to really, really bright, the actual intensity, the pixel intensity doesn't change. It's still measured exactly the same as it was if we left it to the default. And you can turn channels on and off here. Over here are different ways of showing and hiding objects and annotations and things that we're going to add. So there are keyboard shortcuts that if you hover over it, you can see that the A button, so if I draw some annotations, the A button will show and hide the said annotations. You've got the fill button, so if you want to fill the annotations in to make it look a little bit more sort of solid, you can do that. The annotation names, if you've named the annotation, this is just called annotation, that will change and show and hide that. We won't be using microarrays, detection objects. So detected objects, when we start detecting cells, we can show and hide that and make them solid as well. This C button is really good for pixel overlays. This is when we start to train the model and use thresholds and things like that. That will allow us to sort of remove it, because when the threshold comes up, it actually just fills in everything. So if we're still training our model, it can get in the way. So if you hide it, it allows you to then see the data as it is, and then you can continue to train the model without it being so cluttered. The Fiji button, if you want to send things to ImageJ, I should say, then you can send that to ImageJ for filters and things like that. If you want to do extra measurements that QuPath is not capable of, you can send a section. Remember that you can't send the whole thing. You need to select an area and then send the region to ImageJ in order to analyze it. The whole thing is usually too large for ImageJ to handle. So that's the very basics of QuPath. If you want sort of more in-depth details than about functionalities, you can go back to part 1 on our YouTube channel. It's also available in the data folder where the recording and the PDF of the presentation will be found later on. Right, machine learning, object classification. So if you remember back, we did some positive cell detection of an H&E, or of an HDAB image. So we had an HDAB section where we used the cell detection algorithm to detect all of the cells using the hematoxylin stain, and then used the positive cell detection algorithm to detect whether it was positive or negative for DAB. Now that used simple thresholding. Essentially, we used the slider to set a simple threshold to detect whether it was positive or negative for DAB. Now, the object classification is a trainable function which uses real cell data to determine what is and isn't positive or negative. Now, because we are using lots of multiplexed images, it's possible to use the separate channels to train different, to create different algorithms for each channel, and then combine it into a single algorithm to detect the multiple markers in a single tissue. Now, we typically use a neural network to learn, and that's in quotation marks, and make decisions based on your input. This is known as machine learning. So machine learning should be sort of kept separate from artificial intelligence. There is no real intelligence here. It is literally using a mathematical model to determine what is and isn't. It doesn't think for itself. So it doesn't make its own decisions. It literally just does what you tell it to do. Now, most of us have been actually training machine models for a long time. If you've ever seen a capture thing like this, you've actually been helping to train a model. The whole point of this is to get human input to tell the model what is and isn't a thing that it asks you. So in this, select all with trees. You select the things with trees in it, so the model can then build on that to create the model for actual object detection in a grander scheme. So the information is stored as a mathematical model and forms an algorithm that can be used to make predictions and classifications. So if you then, after multiple rounds of this with human input, you then give the model an image that it's never seen before, the hope is that based on the input from the humans, it can then pick and choose what is and isn't a tree. So that allows us to save a lot of time because computers can think a lot faster than us and can run through a lot more data than we ever could. So to make our lives easier. So the result should always be taken with a grain of salt. There is no intelligence here. It's just a probability calculation based on what we have given it. There are two main algorithms that we use in QuPath. The first is a random tree and the second is a neural network. Now, a random tree is literally just a decision matrix. So a yes and no thing. Is it this? Yes or no? Then we go down here. Is it this? We go to the next one, etc., etc., etc. Very, very basic way of doing things. The second more advanced way is to use a neural network, which is designed very similar to the human brain, where you have a lot of interconnecting nodes, which can then make decisions based on the input. So typically with most neural networks, you have an input layer, an output layer, and a hidden layer, or multiple hidden layers. The input is the data that it sees. Now, in this particular example, it's been given a handwritten 2. We know it's 2 because our brain tells us that it's 2. So what happens is this data is then inputted into a whole bunch of nodes, in this case 784. It's then sent through to the hidden layers where a decision is made, another decision is made, another decision is made based on several aspects, and then you get the output layer, which tells us that it is a 2. The computer has decided based on its input it is a 2. So Taylor wants to come in. So both classifiers may give you very different results. I've had the best results based on the neural network. So when in doubt, just use the neural network as it is, quotation marks, smarter. Now, multiplexing or multicolor microscopy is used to allow us to characterize diseases and gain better understanding of biological processes. So if you have a whole bunch of markers that can be used to diagnose particular diseases, for most likely cancer, is very useful with multiplexing. Then you can basically use a whole bunch of these different markers to light up the cells that are expressing these markers and then make diagnostic decisions based on what is actually showing up in those tissues. Now, anyone who's done immunohistochemistry knows that typically you can only do a few different fluorophores with conventional microscopy. However, multiplexing involves using a specific spectral deconvolution system and I've seen them with up to 100 markers. So you can get lots and lots and lots and lots of different signals and different markers from a single tissue rather than the standard 3 or 4 that we would typically do with typical immunohistochemistry. QuPath then has the ability to take all of those channels and learn based on algorithms that we feed it what is a positive and negative cell. Now we've got this image here. We've got these different channels, 1, 2, 3, 4, 5, 6 different markers, as well as DAPI for cell detection. So we use DAPI to determining what is a cell, and then we can use QuPath to determine whether each one of these cells is positive or negative for each of these 6 fluorophores. Now I'm just going to escape from that. The typical default classes. So if we remember classes, if we select a particular cell, so if we highlight this particular cell, we can then class this as a specific marker. Positive, negative, tumor, not tumor, etc., etc., etc. In this case, we can actually mark them with the particular protein marker that we have actually selected. So we can actually go through and mark them individually. The color will change depending on what we're looking at. And you'll see over here in the annotation list, you'll get these different annotations with the classes that are there. Now, rather than putting these in individually, you can actually make QuPath do it for you. So I'm just going to get rid of all of these. So I'm going to select all of them and delete. Can I delete them? Or delete. Yes, okay. Normally the delete key works, but that's fine. So now we have a blank slate. I'm just going to get rid of all of these annotations as well. Now, if you go into the channel tool, we can see that each channel is named specifically. In most confocal images, when you set up your different channels, will typically have the name of your of the fluorophore you were looking at in each one of the channels. So what we can do is tell QuPath to use these channel names to create our classes for us. And what you would do is you would go into where is it? Into here, into this little arrow tool under the class list, and populate from the image channels. And if you click that, automatically all of those go in there. There is always an ignore channel. So it automatically creates an ignore channel just as part of the algorithm, even though there is no channel called ignore. Cell detection. So before we can start telling, before QuPath can start determining what is and isn't a positive cell, we have to show QuPath where the cells are. Now, I feel that there is a little bit of a bug in version 0.6 going from 0.5. Now, typically what you would do is on the original image, before you started splitting the channels into individual training images, you would create your cell detection here and then duplicate the image for as many channels as you wanted to select, and that detection would then propagate to the rest of them. It doesn't seem to do that, at least very well. So you do it on the first image, and then you would propagate it to all the channel, the separate channel images that we're going to create for the training image to save ourselves some time. But it's just, for whatever reason, I can't get it to do it anymore. But that's fine. It's just an extra step that we have to do. So what I'm going to do is I'm going to go back here, and I'm going to create our training images. Now, we typically create a training image for each fluorophore, each channel that we want to train QuPath to detect. So what we do is we go into classify, training images, and create duplicate training channel training images. So if I click that, it's going to give us a list of total channels that we have. Now, we don't need one for autofluorescence because we're not going to train it to detect where the autofluorescence is. DAPI, we're not going to worry about, but we are going to create one for each one. When I click OK, what it does is it creates an exact duplicate of the original image, but it labels it with each channel. Now, these should be all duplicate, and I go here, and it's exactly the same. But we're going to create a different training image with each one. So for this one, I'm going to do CD8. I'm not going to do PDL1 because I'll show you in a minute, but because PDL1 is red, when I try to train it, because all the cells when they are detected are red, it gets a bit annoying when you're trying to train it. But it's not that we couldn't do it. I'm just going to take the easy way out and do it here. So what I'm going to do is I'm going to use my square tool to create an area. It can be as big or as little as you want. So I'm going to create it around this section here. And then I'm going to go into the cell detection algorithm, which is an analyze, cell detection, and cell detection. We've seen this box before. All of these, I'm not going to go over these too much. If you want to sort of work out what each one of these are, you can go back to the original part 1 where we go over them in depth. The pixel size is based on the resolution. The background radius is typically how big the nucleus is going to be. These filters here allow you to do a bit of blurring, etc., to make the algorithm, it fills in holes and things like that. You can fiddle around with that if you want. The minimum area of cells, the maximum area of cells that you want to change. You can, if you're getting a lot of sort of fragmented cells, you can reduce them if you want to, or increase the size. And if you're getting too big of clusters, you can decrease this size. The main thing is the intensity parameter. This is the intensity of the DAPI stain. So here we've automatically chosen DAPI as our cell detection channel. Technically, there should be 1 DAPI nuclei per cell, so that's what we're using. But this is based on the intensity of the stain. Now, by default, it's 100, but I found that for this particular image, 0.5 is good enough to choose to select the thing. You can play with that depending on the sample, but that's usually pretty good enough. Now, remember that while QuPath will detect the cell, the actual margin that it creates is artificial. It's not based on anything, any data that's actually in here. We tell it how big from the nucleus to expand the cell wall. So I'm just going to put 5 microns. Now you could make this as, if you're dealing with smaller or bigger cells, you can change that, but 5 microns is typically all right. I'm not going to include the cell nucleus because I don't care about the cell nucleus. I care about what's going on in the cell as a whole, but you can include that, and then up in view, you can turn off under cell display. You can just show cell boundaries, or you can turn the nucleus on, etc., etc. So you can actually change that, but I'm going to turn that off. I want smooth boundaries just because it looks a bit nicer. The main thing is to make sure that you choose make measurements, because when we start training the algorithm, it needs measurement. It needs a measurement of the intensity of the cell to determine whether the cell is positive or negative for that particular stain. So that's very important to make sure that's on. And then we go run. And then it'll think about it for a little while. And there we go. So if we go into here, it's done a pretty good job. So, I mean, it's missed a few, but as I always say, I'm a 95% person. As long as it's about 95% accurate, better is always, higher is better. But as long as it's 95% accurate, that's typically good enough for me. Because if you really sort of think about it, doing this manually would take so much more time to get a little bit more accuracy on this. So I'm usually pretty happy with this. If we want to, we can turn off the other channels to see exactly how accurate it is. So we can actually just show the DAPI nuclei. We can make this a bit brighter so we can actually see with the nuclei, and it's pretty good. I'm pretty happy with that. We can fiddle around with the cell detection if we want to, but that's fine as is. Okay, we don't need cell detection anymore because we have now detected the cells. Now, if I have a look at the actual image where we're actually on the CD8 image. Now I'm going to turn off DAPI and I'm going to add CD8, which is there. That's the CD8 channel. And if I increase the intensity here, we can see where the CD8 positive images are, and we can see where the CD8 negative cells are. Forget the other channels. Even though they're there, we are not, we're going to tell QuPath to ignore them while we're training this. We're only interested in the cells which have this. So the next thing we're going to do is we're going to go into our training algorithm. So to train the model, we go into classify, we go into object classification, and train the object. Now, these are objects. When it creates these are what's called an object. It's different to an annotation. The annotations are the regions of interests. The objects are the cells themselves. These ROIs that we've trained, they're very different to the annotations. And if we look at the annotation, we can actually see in the annotation that we've created, there are 2056 cells within that particular annotation. So it's different. Annotations and objects are two very, very different things. So classify, object classification, and train object classifier. So this gives us a few options. Now we're going to be detecting, we're just going to do detecting all. You can just say detect cells, but we're going to detect all. Then you've got the option of the type of classifier, and random trees and artificial neural network are the usual ones that I use. K-nearest neighbors, a very dumb algorithm. I think it's below random trees essentially. So pretty much all the time I will use the artificial neural network. For features, now I said before we have created each one of these training images to do a specific fluorophore, and that's where we select the particular features here. So I'm going to do the selected measurements, and we're going to select the measurements. Now, you may get an error here. If you click this, you may get an error that there are no measurements. It's usually because you haven't ticked that measure button. That's where you would fall down with that. So you would need to redo those, delete that and redo it with that ticked box. And that's the error that I was telling you about with, I think it's a bug. Whenever I try to propagate this from the original image, it just doesn't work. It just comes up with a measurement error. So that's why I'm having to do them individually for each one. Now, this gives me all of the measurements that it has created from all of the cell data. So area, perimeter, circularity, mean intensity, standard deviation for the fluorophore themselves, the fluorophore in the cytoplasm, and so we're going to be using this. Now, what we're going to do is we're going to filter firstly by the channel that we're looking at. Now, if you, for whatever reason, you've accidentally covered this, you can't move these now that this is thing, but if you've actually covered that, you can actually look up the top and you can see I'm on CD8. So I'm going to type CD8, and this will give me all of the CD8 information. So I'm going to select them all. Don't click apply yet, because just remember there is DAPI and there is autofluorescence in there. So I'm going to filter by DAPI, and I'm going to select all, and I'm going to go to autofluorescence, and I'm going to select the autofluorescence as all. So our detection is going to be based on the channel itself, the CD8 channel, the DAPI channel, which is going to be in the nucleus, as well as the autofluorescence, which is part of the image. So now, again, don't click apply. What you want to do is clear the filter to bring everything back, and then if you cycle down, you'll see that only the things that we've selected are now there. If you select it with the filter on, it will only select those and skip out everything that we've already done. So make sure you clear that filter, and we click apply. Okay, so now we're ready to train the model. Don't click live update yet. Live update, if you start doing live update, the train object classifier needs at least 2 separate classes in order to make a decision if something is or isn't. And in basic terms, you need the channels class. So in this case, we need the CD8 class, and we need an ignore class. That's typically the basics for this. So first of all, I'm going to come in here, and I'm going to use an ROI to start telling it what is and what isn't. And you can actually see there is some background here. So we want to make sure that it's not including the signal, which is just basically it might be non-specific staining or autofluorescence, etc., etc. So you've got several options here. You can use any one of these ROIs. I find it easier just to use the spot algorithm, the point algorithm. So I'm going to create a new class, and I'm going to set this as CD8. And I'm just going to go through and start selecting cells which have CD8 in them. Now, how many do you need? How big is a piece of string? That's the question. More is usually more. As many as you can get is usually pretty good. But I mean, technically you need one. That's all you need. But the more you add, the more specific it gets about the training. Now I've got 15 points there. I'm going to add another one, which is my ignore class. I'm going to tell it what to ignore. Now that's probably negative. It's possibly positive, but I'm going to treat that as negative. I'm going to treat that as negative. It's always a good idea to have negative channel around the cells that are positive, because you get some overflow from the positive cells. Now that I have actually done two separate channels, I can now start my live update. And you can actually see the red ones are negative because those are the ones that I've labeled, as well as what the other ones are sort of being determined at. Now, I've got a positive cell here. Now, that's probably negative, so I'm going to go and make sure I select my ignore class, and I'm going to make that ignore, and that should then turn red. That's probably positive. I'm going to leave that as positive. But what you want to do is look around and make sure there are no positive samples that are obviously negative. That's probably positive. I'm going to leave that as positive. That's definitely negative. I'm going to say that is negative and then turn that off. And by doing more and more of that stuff, you build up the model to be more accurate. That's probably positive. So I'm going to go in here and make that a positive. There we go, and turn that on, and etc., etc. Now that's probably good enough. So now I've trained my algorithm to detect CD8 in these images. I'm going to go back to my train object classifier, and I'm going to name and save my classifier. So this classifier is CD8, and I'm going to save that. So now that's saved in my classifier in my project, and that classifier can then be used for any other image in my project. All right, so I'm going to close these, and now I'm going to go from CD8 to another channel. I'm going to go to CK, which is the blue channel. So do I want to save the changes? Yes, you want to save those changes. Now, the same channel's being turned on, so I'm going to turn that off, and I'm going to turn on the CK channel so I can see the specific channel that I'm looking at. Now, these are large clusters of cells, so rather than using the point tool, I can use things like the paint tool. So first of all, I need to select an area, again, do my cell detection. The only thing I really need to change here is my threshold, which is 0.5. Now, even though DAPI is not selected, it is still there. It's still hidden, but it is still a selectable channel. It is still being used for the detection. So I've got a few options here. I can use the any one of these. Sometimes it's easier to use the magic wand tool. So if I use this, I can then use the magic wand tool to select specific areas based on the intensity of what I'm looking at. Now be careful not to choose dead space, because so you don't want to go over into here, because that will obviously make a positive a negative. So I'm going to select that as CK, positive. And I'm going to do the same thing with the negative space. So I'm going to come in here, I'm going to choose, delete. I'm going to choose the square tool and just select this entire area as an ignore class. So now what I can do is I've now labeled two classes, minimum. I'm going to go into my object classifier and start training my object classifier. So you can add those annotations at any point. So again, I'm going to go into artificial neural networks. I'm going to go into selected measurements and select the measurements that I want. Now this time I'm using CK. So I'm going to go CK, select all. Now have I got a problem? No, that's fine. CK. And I'm going to go into, again, DAPI and select all, and then autofluorescence. Oh, autofluorescence and select all. And then wipe out my filter. And then you should see those particular objects selected, and I'm going to apply that. So now I should be able to go to Live Update, and then automatically we see all of those blue cells being selected. Here, I'd probably start bringing up my point tool, and I'm going to add an ignore class. And I'm going to come through, and I'm going to start labeling ignore, ignore, ignore. Any cells that are probably, now that one's probably positive, so I'm going to add a CK class. So I'm going to come over here and say that's positive, that's positive. Now, there is an issue there. It might be because I've selected a bit too much dead space in the over here. So if I get rid of this large thing and be a bit more specific, there we go. That's better. So yeah, that's why you don't do dead space, is because you can run into problems by selecting that dead space. You can actually run into problems with it then propagating to other areas. So here, that's probably positive, but that's mostly negative. So I'm going to go ignore, ignore you. Spot. There we go. Spot. There we go. And again, more is more. The more you add, the better your algorithm is going to be. For some reason that's, there we go. Yeah, so that takes care of those. Keep going around, adding more and more, and you'll find that the algorithm will get more and more accurate. So I'm happy with that. So I'm going to close that down, and I'm going to give this classifier a name. This is CK, Opal 690, and I'm going to save that as my classifier. And I'm just going to do one more. I'm not going to do them all, otherwise it'll take too long. And I'm going to go to, let's say, FoxP3. So I'm going to go to FoxP3. I'm going to turn that on. And I'm going to go to my FoxP3 image here. And remember, when you move away, save the image that you've just changed. And then you'll get a blank slate with the next lot of images. You then create your area, do your cell detection. I'm hoping in a new version that they'll fix that issue. You should only really have to do it in that first image. And when you create the training images, it should then propagate into the new images. For whatever reason, it doesn't. I don't know why. And I'm going to go into cell detection, change my threshold to 0.5, run. And there we go. This I'm going to use my spot, my points here, create my two annotations. One is going to be FoxP3, and this one is going to be ignore. So I'm going to go through and I'm just going to label some FoxP3 cells. That one is probably a little bit inaccurate. There we go. And my ignore class, always good idea to do them around positive images, just to sort of deal with the potential overflow from the channel. Now I can then bring up my classifier, my object classifier, train object classifier. I'm going to choose artificial neural networks, my measurements, selected measurements, select. Now I'm doing FoxP3, select all, DAPI, select all, autofluorescence, select all, just clear the filter and apply. So now I can do my live update, and then automatically you start seeing these cells are turning orange and the red ones are ignore. Now, there's an issue there. So I'm going to go here and select that as a positive cell and that change that. And that's pretty much all there is to it. You can do that for as many channels as you need to. Um, and more is more. The more you do it, the better the algorithm can get. Obviously, if you're making too many mistakes, that can lead to problems. So yeah, why is that negative? No. Oh, wait. I made a mistake there. Now, if you want to, you can turn these on and off. So you can turn off the ROIs. You can turn off that if you need um, to sort of clear that out and see sort of where you are. Okay, so once I'm happy with that, I can come in here and go and save this as Fox P3 and save. Okay, so now I have built up at least, I think, 3 separate classifiers. I could take any one of those classifiers and then apply them to the images. However, I want to do all of them. If I wanted to build up all 6 of these channels, that is fine. Turn on all the channels again. But now I'm going to go back to my original data. And so now I want to apply those classifiers to this original image. Now, I could do them individually, but I want to do them as one go. So what you would do is you would combine them into a single classifier. So to do that, we go into classify, object classifier, object classification, and create composite classifier. And that will show me all the current ones that I've saved from the other images. If I'd done all 6, then I would have all 6 available. And I can just either individually or all together. And I'm going to give this a name as composite classifier. You can call it whatever you want, as long as you know what it is, and save. So now I have created a new composite classifier as well as my individual ones. So now I'm going to use this model to classify this image. So I'm going to do an image. I'm going to do a ROI. I'm going to do an annotation across the entire image. I'm going to, first of all, of course, we're going to go into cell detection, threshold down to 0.5, and make sure my measurements are on and run. It's going to take a little bit longer because we're doing the whole image. Now, it's not showing up. Why is it not showing up? Because I have turned off those annotations. If something doesn't show up when you think it should, it's very likely you've turned it off. So you need to turn on your annotations there, your object classifier. Okay, so now I've got a blank slate that I can now try and use my classifier on. So what we're going to do is we're going to use the model by going to classify, object classification, and instead of training the object classifier, we're going to load the classifier. So if I click that, it gives me my individual ones as well as my composite. And then if I apply that, here we go. So now all of these images, all of these cells have been classified specifically for the channels that we have looked at. Now, we only looked at CD8, CK, and FOXP3. There are other channels in here that are in there that would be classified if we had bothered to do the classification. So you can do as many channels as you want. Now, you may find that some cells are actually multicolored. So some cells actually might have a weird shade of yellow or green. It's because it's got multiple markers. Now, if we have a look at this, this is a weird sort of shade of green. So if we start removing some of these channels, we may find So that's positive for yellow. It's also positive for orange as well. So you will get sort of combinations of channels within a single cell, and it won't be a perfect blue or red, but it'll be a combination color. And that tells you that there are multiple markers actually, rather than a distinct single marker. Results. So now we have this image, and we can export this image if we need to. You can fill in this using that. If you want a little bit more, something a little bit more striking, you can use that function. Oh, Taylor's left and come back. There she is. So you can turn this on and off if you want something a bit more striking, or you can just use a simple fill in. You can add the nucleus back if you wanted to. So if you come in here and go view, cell display, cell and nucleus boundary. So there's your nuclei. You can just do the nuclei if you really want to. So if you change this to just nuclei, it's up to you what you want to do to display that. And right, so now we've got a picture. We need quantification. We need some sort of results that we can then export to a spreadsheet. So I'm going to turn on cell boundaries. Okay, so if we go into the image, annotations, I should say, and we have a look down here, we can actually see some of the values. So in this entire image, we've detected 5,282. And we've got the individual numbers for each of the channels that we have detected. And you can actually see there are some individual ones. There are ones with multiple channels. That one has all three, but that has two, that has two. There are none with all three, but there are a couple with duplicate markers. Then if we go into the detection measurement, so if we go to the measurement tab up here, and go to detection measurements. Give it a second to think about it. We get the results table, and it shows you for each and every object that it's detected, the actual results. So we've got a whole host of results. I'm not going to go through all of them. There are hundreds there. But it shows you the intensity, the standard deviation, the mean, the mean, the max, etc., etc., etc. What it was used to detect these. It gives it a classification. It's only positive for CK, the blue channel, this first one, and then it gives you the results for each and every measurement that we've done. You can then save that as a spreadsheet. Results data is an Excel spreadsheet. I think it's a comma delimited file, but it will open quite happily in Excel for then to do your statistical analysis. And that's basic. That's the basics of doing of training an algorithm and then putting it onto a data set. Now you can then use that that classification for any data set, which has similar images. Now there may be variations amongst your samples. So it can be difficult to get a working model off a single image. Now we've used the same image to do detection in that exact same image. What you would typically do is train the model on multiple images, multiple data sets. So 5, 6, 7. Again, more is more. The more data sets you use the training algorithm on, the better it gets. Now, it's not a magic bullet. Just remember that it is not a magic bullet. It is a mathematical model that's only as good as we really train it, as good as our model data is. Now, QuPath, the QuPath website literally has a section on the website called Knowing When to Quit. Knowing when to quit is that if your data set is not up to par, like if you've got obvious artifacts with your staining, if your imaging wasn't good enough, etc., etc. Basically either you can just do it manually, or go back and do it again. Make sure the data is actually up to scratch for training the model. Because remember the old adage, garbage in, garbage out. I mean, the images that I see coming through the facility are amazing. So I don't think that's going to be a lot of people's problem. But just be aware that if you don't have the data set that is up to scratch for training the data, training the algorithm, then you can run into issues. So we used single images, this single image, to train our model. How do you use multiple images? Now, what you would do is create a training image. You would load up all of the, like, however many images you want to use for training, and then you would combine that into a single image. And it's pretty easy the way that it does this. So what you would do is go into, where is it? Into classify, training images, and create a training image. Now, what you would need to do is to go through the image, go through all of your images, select areas within each image, and then the QuPath will take every one of those areas and combine them into a single image. So what I'm going to do is I'm going to hide this. Can I hide it? I'm just going to delete it. I really need to. I can redo it if I need to. So I'm going to delete all the. No, I'm done. So now I've got a blank slate. So what I'm going to do over here in the class list is I'm going to create a new class, and it's called training data. Now, if you want to change the color, you can just single click, I should say. Give it a color that you're going to recognize as, I'm going to do this as, I don't know, dark green. That'll do. So what you would then do is go to the area and select the area and set that as a training data. So now throughout all of the images, when you run the algorithm to combine all of those sections, what it'll do is it'll crop out that, will just take a copy of it and then merge it into a single image. So you can add as many as you want across every data set, training data, blah, 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 blah. You can also do this a little bit more randomly, not so subjectively. So if I get rid of this, you can go into, where is it? Classify, training images, create region annotations. There we go. So then you can go and say, I want to create a width of maybe 200 by 200. I'm going to classify it as training data, and I'm going to do it randomly. And then I click create data. There it is. Create data randomly across an image. Then you would save this image and go to the next image data set. So you just keep repeating that over and over again in multiple training data sets. Once you're done, you would then create the training image. So you would go to training images, create training image, and you would select the classification that you've created down the bottom, training data. You'd create a data width. I think 50,000 is a bit too big. So I'm going to create one that's maybe 4,000 pixels maximum. It's not going to, if there's less than 4,000 pixels worth of these images, it's not going to just create a 4,000 pixel image. And then I'm going to click OK. No suitable annotations found in the current. I've not saved it. Okay, make sure you save it. Okay, let's try that again. Training image, training data. Okay. There we go. So now it's created based on those. Yeah, so make sure you save it, otherwise it doesn't know that it's there. It's now created those ROIs into a single image. Now, if you had dozens or hundreds of these images, you'd get a massive image made up of all of these sections. I'm going to say that it's fluorescence, apply. And then again, what you would do is do what we did before and create a ROI. Of course, DAPI is there. It's just not showing up. And then do your cell detection. Cell detection. Oh, sorry. No, first of all, what you would do is you would create duplicate channels. So you create duplicate channels to propagate that out into separate channels for the algorithm. Then you would come into the, for each one of them, you would go cell detection, set your settings, and then go through the workflow as we did before. For each one of the channels that you want to detect, run the training algorithm and save it. And that's all for the object classifier. So we're going to go on to B, the pixel classifier. Very, very similar, but instead of looking at individual cells, we're just going to look at the data set as a whole. So the object classifier versus pixel classifier. So the object classifier allows us to detect specific objects. In this case, in the case that we looked at, these were cells, and then detect them and then create classifiers to determine if they're positive or negative. But we can use the pixel classifier to detect specific areas and objects with a defined signal. Tumor markers, non-cellular objects. These don't have to be tissues. These can be photos of things. So if you have a photo of a gel, or maybe a photo of a petri dish of bacterial colonies, things like that. So you can use it to detect actual objects within an image. Now, the pixel classifier doesn't need to use detected objects like we were for the cells. We can use the image itself to determine the areas we want to detect. Now, it's very important, remember to color inside the lines. If you are selecting the area that you want and then you go outside into the background, that gets taken into the model. So you can run into problems with it with an incorrect detection. So we're going to train the object detector very similar that we did before. The neural network usually gives the best results. And you would use an ROI to highlight a specific area that we're going to be using to detect the signal, the pixels that we want to detect. As before, we need at least two classes. We need the thing that we're looking for, as well as at least the thing that isn't what we're looking for. So in that case, you would do the object, the particular part of the image that we're looking for, as well as the background, which would be the negative. However, you can then do this particular area, this particular type of object, and another different type of object, as well as another object, and then the background. So you can do multiple, select multiple objects, multiple different signals in the image. And I use the live update to determine what is and isn't the particular area. Now I'm going to do something a bit less tissue based. I'm going to, if I can find the image that I had before, that's it. Drag and drop that into, it's a bright field, I don't know, it's an other, I suppose. Yes. And here is a whole bunch of flamingos. So these are, what I want to do with this image is I'm going to determine what is and what isn't a flamingo. And that's what I mean about determining pixel values. We're looking at specific pixel values, which are the thing that we're interested in, as well as the things that are not. So what I'm going to do is I'm going to bring up instead of using the object classification, I'm going to build up the pixel classification, and I'm going to train my pixel classifier. I'm going to use the artificial neural network as normal. Resolution, I use the resolution firstly using usually moderate. Now, what you'll see in very large images is as you as you train or run the classifier, you'll see it propagate out from the center. Now, if you have a very large image and you're using very high resolution, it takes a lot of computing power. But if you use a lower resolution, if you downsize it even further, what happens is that the computer doesn't have to think as much. So once you have a really good algorithm, you can then turn it up to a higher resolution rather than every time you add a new area, it's continuously trying to find it. So I usually use a moderate one. Features, the features are based on the channels. Now, this is an RGB image. So there are 3 channels, red, green, blue. If you had a fluorescent image, it would be whatever channels, DAPI, GFP, whatever are there. I typically use for features, you can use different filters to aid in the analysis. Like what it does is, in the case of Gaussian blur, it blurs the data into a more, I don't know, palatable format that the algorithm can actually look at. Unless you can turn this off, but I find that turning it off uses a bit too much computing power. But if you blur it a little bit, the function works a lot better. Now, I'm not going to go into all of these because some of these I actually don't know what they do. Gaussian is typically, when we're doing sort of feature extraction, Gaussian is usually the 9 times out of 10 I'd use a Gaussian. Laplacian and all these other things, Hessians. I've used Hessian filters before, but it depends on the structure. So I typically use a Gaussian filter for this. These I'm going to leave as none because I honestly can't remember what these do. The local normalization scale, QuPath, this is from the website, generally best avoided. This can optionally be applied to some local background subtractions and normalizations in an effort to handle image variations, but in practice, it often does more harm than good. Maybe removed or replaced in a future. They're actually thinking of removing these from future versions because they actually do more harm than good. So I'm going to, of course, red, green, blue. The scale, now scale is the full resolution. So 1 is the full resolution of the image, but you can actually take this is similar to the resolution. You actually take the resolution down. So I'm just going to leave that as 1. I'm going to, based on classification rather than probability. Probability is more of a mathematical thing rather than the classifier that the neural network uses. So just use classifiers. And I'm going to do everywhere. Now, if you wanted to, you could just select an area. So if I wanted to select an area, I could just do any annotation and it would only look at this. Now, that's a good idea for large data sets. If you have a massive data set, and I've done this on slide scanning images, and what you see is often to try to do the entire image, you see it takes a long time. You see the pixels sort of like propagate out from the center. But if you wanted to speed things up, especially in the sort of the early stages of doing the model, you can just do a small area. We do this in Amaris all the time. We would crop down the area that we're looking at. But for this image is fairly low resolution, so I'm just going to do everywhere. Okay, I'm not going to turn on live prediction right now because I haven't added any of my classes. So I'm going to bring up my classes and I'm going to get rid of everything. Just delete, remove. And I'm going to create 2 new classes. One is Flamingo. And one which for some reason has labeled it pink. And one is going to be ignore. So now I'm going to go through and start labeling my image. So first of all, I'm going to use, I can try the magic wand tool and start labeling with the magic wand tool like pixels. Being very careful not to go over into the background. and I'm going to label that as flamingo. And then in the background where the water is, I'm just going to select an area and label that as ignore. So now I have two classes. So now what I can do is turn on the live prediction. And you can see here that it is already determined that almost 50% are ignore and less than 50% is, less than 50% is actually flamingo. Now, what I should be seeing is all of the objects being labeled. Now, the reason for that is I've got the C button turned off. Show hide for pixel overlay. So if I turn that on, then you start to see where it's determining that is. Now, this is very pixelated and it's very non-specific right now because first of all, I haven't chosen many areas, and second of all, I've got the resolution turned down. Now, if I turn the resolution up, there we go. Automatically, you see flamingos. And if you sort of turn this on and off, you can automatically see flamingos. So I'm going to turn this to very high, just so it's a bit, actually for this, I think I can just leave it at full. So you can sort of see where the flamingos actually are right now. Now, the reason we have this button is now if I want to try and optimize my model, it's very difficult because I can't see the flamingos anymore. I can turn this down to change the opacity, which can help with that. So if I turn that, or I can just turn it off straight away. So what I'm going to do is I'm going to look for troublesome areas. Now, I'm not too keen on trying to do the legs because if I try to do the legs, it might cause more problems than solve, but I'll give it a go. Let's see if I can find the legs. So what I'm going to do is I'm going to use the paint tool and I'm going to go very high resolution to try and get as much of the legs as I can. But what I might actually find is because the legs are very, very close to the color of the background, it might actually cause more problems than it solves. So what I'm going to do is I'm going to label this as flamingo, and actually that did a really good job. Here we go. So it's actually started detecting the legs. So that's actually better than I thought it would be. So if I turn up the opacity, we can see a lot of flamingo legs. Now I'm pretty happy with that. That's really working quite well. If I wanted to, of course, I can go through and start labeling ignores, etc., just to sort of get a little bit more data in there. But as it is, I think that's a winner. There's a few there. I probably wouldn't bother with them. It's a bit too... Actually, I think I ruined my model right there. Did I? Oh, no. That's fine. So these sort of closer ones, the ones that are closer to the camera, yeah, so I think by adding some of these other ones, in this case, less is more. So I'm actually going to delete that one. And has that fixed my model? Yeah. So I think I chose an area where there were very similar pixels to what was actually in my flamingo legs. So if you make a wrong turn, just simply delete the annotation and that should fix the issue. Okay, so what can I do with this image now? If I wanted to, I can then save this classifier. So I'm going to call this flamingo and save this. So any similar images, I can then use this model to detect flamingos in other images. So what I'll do is I can so save this. Yes. Now, if I delete all of these annotations, delete, yes. Now that I've created my model, I can then apply it to this image. So what I'll do is go into classify, pixel classifications, and load the pixel classifier. Choose the model, flamingo. I'm going to do this everywhere. And you can automatically see that it has done my detection for me. Now, how can I do some measurements for this? What you can do is over here is create objects. Now, remember, objects are what we used previously for detecting cells, but they're for any object that we want to do analysis of. You can use that for that. So I create the objects on the full image, and brings up this thing here. So I'm going to create annotations. I can filter out smaller objects. There doesn't seem to be any sort of little annoying objects, but I can say I want to filter out anything that's less than 10 square pixels or things like that. Hole sizes, if you find objects with unthresholded hole parts, like if you see sort of holes in the annotations, this is pretty solid, so it's pretty good. You can filter out holes and make them more solid. Split objects is when the objects are created, instead of them all being housed within a central annotation, you saw sort of one annotation with as many objects, it actually gets split into separate objects automatically. You don't have to do that. And then I go, okay. And now we can see a whole bunch of ROIs have been drawn around all of my objects. So if I turn off the signal, we can actually see all of these ROIs around the objects. There we are there. And then we can go into show annotation measurements. And we have a whole bunch of measurements. Now, that's based on the annotations. So what we'd have to do is, I think you have to measure, show detection measurements. No. Add measurements. There we go. So add measurements. I need to do my measurements. And measurements. So where are our objects? So it shows me that this is the entire image has 83 square pixels of area. And I think what I have to do is to split these. Split them. All right, I'm going to start that again. Okay, so let's go to cell detection. No, so pixel classifier, load pixel classifier, flamingo. Yep. Create objects, full image, split objects. There we go. Okay. And there we go. So now we've got individual objects. Some of the, obviously in the background, it's hard to separate objects. And each one of these will have a measurement. So if we go into measure, annotation measurements, there we go. That's what I wanted. So in order to get every single measurement, you need to split the annotations so that each one of these can have measurements. So you've got, just like before, you've got area, perimeter, etc. QuPath, as we would have discussed in the part 1, QuPath's measurement quantification is actually very limited. You typically, for shape factors and things like that, you typically can only get area, perimeter. If you're doing the object detection with the fluorophores, the multiplexing image, there's some intensity measurements, but that's pretty much it. Fiji has many more statistics. Circularity is one that I really wish that they would put in there, although I think you can calculate circularity for the perimeter and the area anyway. But things like Forster, not Forster, what is that? Just trying to remember offhand what the particular measure is, but the longest and shortest axis of an object, you can't calculate that here. So if you sort of had an object, Fiji will actually calculate the longest axis and the shortest axis in X and Y. It doesn't do that. So if you wanted to do that, you have to export this into Fiji to do it there. But from a sort of a object detection point, it is fantastic. It really does a good job. So that's it. That's all I had to talk about today. Now there is the ability to, and I haven't investigated this too much because I'm Fiji scripting through and through, but if you want to, there is actually a way of writing scripts. So you can actually automate with the script editor. So you can create scripts to do analysis in bulk. I haven't investigated it there because I do most of my work is done in Fiji anyway, and I haven't gotten around to it. But I think I think the coding language is very similar to Fiji anyway. You just need to sort of know what the code for specific functions is in QuPath. So if there are any questions in the audience, no questions.
Hey Chad, I do.
Yeah. Hi.
So you're saying that QuPath doesn't have as good of a statistical analysis. Is there any way to like do all of this here and then take it into Fiji, or they're not compatible at all?
Yeah, so it actually has Fiji built into it. So you got image
Oh,
J.
okay.
Yeah, so what you would do is you would send the region. So you would send these objects into Fiji.
Okay.
It's actually
Oh,
image J. So
yep.
it has. I really wish they'd update this to Fiji, but it's image J. But image J itself is still pretty powerful. So you send your object. Yeah, there you go. So it's sent it into. That's a screenshot. That's not what I wanted to do. The first time it does it, it takes a little while to load this up. But once image J is actually loaded up, you can pretty much just do it anytime you want. So you'd send the region. Resolution. I usually use full resolution unless I'm sending a very large section, because otherwise Fiji just can't handle it. Include the ROI, and then you send that off in there, and there's your analysis. So then you can use that as part of your analysis and do all of your measurements. Ferrets diameter, that's what I was trying to remember before. So you can do all that measurement in Fiji rather than QuPath. But QuPath, for most people, are typically generally interested in area intensities in QuPath because it's usually for pathology. But if you want anything sort of weird and wonderful that ImageJ or Fiji can do, then you can send it off there.
Okay, cool. Thank you.
No problem. So speaking of coding, I will be running my my annual Fiji coding workshop. It's part 4 of the series of Fiji workshops that I've been running this year. So that'll be end of November, so in about a month's time. So it's good to have to at least have gone through part 1 of the workshop. However, it's not a requirement. As long as you sort of have a basic understanding of Fiji, then you're welcome to attend. I start the coding workshop with bare basic principles. So if you've never done any coding, if you've never done any coding before in your life, we'll start from basic principles. And I think I've set it for about 3 or 4 hours or something like that. So over the period, you'll build up. Over that workshop, you'll build up a workshop from scratch. You'll build up a script from scratch to automate any work that you want to do, bulk imaging, bulk analysis. So I'll be sending out probably in about a week or two, I'll be sending out an email looking for participants. So you're more than welcome to come to that. But if there are no more questions, I might finish half an hour early. Excellent.
Thanks, Chad.
No problem. Thanks for attending.
Thank you.
Could you create a new classifier for the cells showing mixture of channels? So what do you mean by that, Stefan? Sorry?
So when you had the ones that were kind of a shade of both colors, so multiple channels being expressed, could you create a new classifier for those ones, or is it better to do something else?
No, you could. So what you would do is, let's just do one that we haven't done. I don't think we did PD1. So we'll go back here. What you would do is when you are so you've got all your channels. So when you how trying to put my mind back. What you would do is for the training image, you would create. Sorry. There we go. Train image classifier. What you would do is in the measurements. Oh, okay. So first of all, I have to correct myself. So just give me 2 seconds to do that. Cell detection, DAPI 0.5, run. And then you've got your cell detection. which has now disappeared because I've turned it. Oh, no, it's there. Okay, it's just brown. So now you've got your cell. So what you would do is when you do your object classifier and you select your measurements, now we've got it. So instead of just doing, so for this case, it would be PD1, select all, DAPI, select all, and autofluorescence. You would then choose the second fluorophore that you wanted to add to it. So in this case, I'm doing PD1, so I might want to do PD1CK. So then I'd add CK to the mix. So now when I wipe that out, I should have PD1, CK, DAPI, and autofluorescence. So then what I'd have to do over here is create over here is I'd have to create a new classifier, which would be PD1/CK.
Yep.
And then what I'd do is I'd find the cells which are positive for both PD1 and CK and mark those.
Okay.
And it would use both channels as well as DAPI and autofluorescence to create that algorithm for you. So you probably do it as a separate color that you haven't used before, whatever it is, and you just do it exactly the same way.
And threshold might be different as well for that one, or
what was it? Sorry,
really matter. Threshold, like the intensity, because some of them,
so I mean, it
fainter. Will it still be able to do it?
Yeah, absolutely. So whatever, so it'd be up to you. So if I then chose PD1 and CK, there we go. So obviously that's positive. So you do that as a positive cell. So that would be positive for both of those channels. And then you'd go through, and choose the cells like that one, and then you do probably that is ignore.
Yeah.
So any cells that aren't blue and green, so you do that as ignore because they're one or the other. So you go through and do it like that. But you can do that with as many channels as you want. You just include the measurements for those particular channels as part of your algorithm.
Okay, cool.
And ignore everything else.
Yeah.
But yeah, you'd have to be careful with that because you're juggling more balls than normal.
Yeah, it might take more training.
Yeah, it'd be just as much training. It'd just be you paying attention. That's the main thing. It's just making sure that you're actually choosing green and blue.
Yeah.
In what is positive and sort of like, that's green and blue, but it's probably negative. It's probably autofluorescence, etc. So yeah, you could definitely do it with as many channels as you wanted.
Cool.
Yeah.
Cool. Is there a way to use both the pixel? Sorry.
What was that, Stefan?
I was going to say, can you combine the pixel classifier and the object one into one model? Is that
the pixel classifier and the object? What was that? Oh no!
Can you put them into one? Say if you wanted to do finer objects as well as identifying cells with clear staining.
No, because they're two separate algorithms.
Two separate ones. Yeah.
Yeah, you got these are quarantined from each other. Yeah. What were we saying, Taylor?
I was just going to ask, on top of that, is it possible to train it to do high versus low expression for a double positive? Or is that getting a little bit tricky because it's going to be looking for the same colors?
It's not colors. You've got to remember that we don't colors are like a fake thing. It's all about intensity. So the short answer is yes. Okay. So what you would do is probably you'd probably do two separate classifiers. One, so for green, for example, you would set two classes, one for bright green and one for dark green. So you'd go through and you'd label all of the bright green ones, save that classifier as one color, and then you'd create a new training classifier and go through and label all the dimmer ones. I've never tried that before, but in that case, if you were after that, you're probably better off actually in the positive cell detection, because remember, you've got the different thresholds. So if you wanted to, you could set low, medium, and high intensities. It's threshold based rather than that machine learning. It's literally looking at intensity. So if that was your sort of the way of doing it, you probably do it that way. It's a lot dumber than the object classifier, but that's probably and you turn off single threshold, so you use all three of those. So anything below that is negative. Anything between that and that are low. Anything between that and that are medium, and anything above that is high.
Oh, okay, cool. Yep.
Yeah.
Cool, thank you.
No problem. All right. Thanks for coming. So I will save this. So it should be recorded by Teams. I'll drop it in the folder, and I'll upload it to YouTube at some point.
Well, thanks so much, Chad.
Thanks.
Thanks, Chad.
See you around.
Thanks.
No
Thanks.
problem.
Bye.
Bye.